he discovered the layer. We exposed the layer as a giant hole in the ground. Well, it looks like a giant black hole in the ground. And over a period of time, worked those layers. And it took about another year until Steve Gatesy, a colleague of mine from Brown, was picking at the rocks and found this. And I don't know if you could see it, but here's rock. And you can see this little V-shaped thing here. As soon as we saw that, we knew we found what we were looking for, a flat-headed fish. And as soon as we pulled that out, we found more of these flat-headed fish. About 4 we found that season, and subsequently about 20 individuals, fragments and bits and pieces. They come home on the bottom of a helicopter encased in plaster, and when they come home to the lab, the plaster's opened up and the preparators begin to etch away at it. And here you see, after about a month of picking away at Steve's specimen, the preparator prepared at the top of this thing. It looks like a skull. There's one orbit here. There's one orbit here. This is in the fall of 2005. About 4 months later, this was what was exposed. You see we have a flat-headed fish with one orbit there, one orbit there. Looks like we have a shoulder girdle and, indeed, a neck where the head is separate from the shoulder girdle. And just to give you a sense, after about 8 months of this preparation, here's the fish, here's a tetrapod, here's the new fish. And the idea is a Tiktaalik, that it has scales and fins with fin webbing, but like a tetrapod, it has a flat head with eyes on top, a neck, a mobile neck, and then aspects of a tetrapod limb with elements of the shoulder, elbow, even parts of the proximal and distal carpus. Now, Tiktaalik, when I was listening to Eric Lander talk yesterday, he was telling about the amount of data that genomics is generating. And it was truly daunting to see what he was doing with the amount of data that's coming out every day, every month, every year. And I was excited, and then I didn't know whether to laugh or cry, because I realized I've spent 6 years collecting one data point. But the difference is I could actually carry my data point around, and this is casts of Tiktaalik here. And I brought it so during the break you can look at it. But one thing about something like this is it's an object. It's a physical object. And just to do a minor slight digression, is that the physical object, has a power to it. And it's revealed to me when I visit schools. I do a lot of elementary school talks. And it's almost invariable when I'm dealing with kids between, say, 6 and 9, that they look at the thing, and to watch them interact with it, I ask them, I put it down in front of them, and I say, what is it? It's invariable. One kid says it's a fish because it has scales and fins. And then it's almost invariable that another kid says, no, it's a crocodile because it has a flat head and because it has this crocodile-like head. And then they look at it for a while, and usually one or more say, well, God, maybe it's both. And, that's my entree to say, well, kids, maybe there's evolution, that kind of thing. But it really is the power of a physical object, and that's why museums are so rich. Anyway, over a period of time, we collected a number of these skeletons and began to prepare out the fin. And here's a humerus that was prepared out. It took about 1.5 years to prepare out the fins, but that we did. Here's the pattern of the bones when you remove the fin webbing. And then when you crack open the bones, what you see are the shoulder, the elbow, and proximal and distal carpus of the proto-wrist, if you will. I mean, we can actually see the articular surfaces of these things and to begin to assess how this thing functioned. Well, that's great, and that's wonderful, but that's not where we're going today. Where we're going today is the questions, and new and powerful questions, this gives us a chance to ask and hopefully answer. That is, we now know this about the fins. We can now ask the question, what does this tell us about extant fins? And what experiments does this suggest to us in developmental genetics? So now we're going to go from the expeditions that are targeted to key parts of the life, the tree of life, finding Tiktaalik and others, and now looking at the developmental genetics of non-model organisms. And what do I mean here? Well, let's just look a little bit at what a tetrapod limb is. It has 3 components, broadly speaking. 1 bone, 2 bones, and then the wrist bones and the fingers. If you look at these in the development, as many have, Zenita Boole, Cliff Tabin, and others, what we see are 2 broad phases of Hox gene activity that are associated with the pattern specification of these components of the appendage. So there's this Hox phase 1 expression, where you have these nested patterns of expression of different paralogs of the Hox D and A cluster genes. And then when you misexpress or you knock out these genes in Hox phase 1, what you do is preferentially affect this stem of one bone, two bone area. Then when the specifying the wrist and the finger bones, there's another phase of Hox gene expression that comes on. It's presumably independently regulated, and this is called Hox phase 2, which consists of a distal phase of expression. When this is manipulated, it preferentially affects the wrist bones and the digits. And the theory at the time that was proposed by Tabin, de Boule, others, including myself, was that really the origin of appendages, vertebrate limbs, is associated with the origin of Hox phase 2 expression. Now, when you look at the fossils here, you know that that can't necessarily be right. And in fact, it suggests something to look at. That is, what you see here is there's one bone, two bones. I'm just looking at Tiktaalik here. And you have a whole bunch of stuff in a distal or third compartment. The question to ask is, are there elements of this phase 2 already established in fins that enable that distal portion to develop? And so what we decided to do was look at an extant ray fin fish because of its position in the cladogram and also its morphological structure, and to see whether it has Hox phase 2. And this is Polydon. It's a caviar fish, like a sturgeon. We don't eat our lab animals. We let them develop. But here's a grown-up Polydon. This is its fin. And the idea is there a Hox phase 2 expression here? Marcus Davis in the lab a few years back worked on this. And you could see in early development, what you have is a classically nested pattern of expression of Hox phase 1. And then coming on later in development, when the fin is much larger, is another distal phase of expression. It's not identical to tetrapod limbs, but it's definitely there in every case we look at. So the idea here is then knowing something about these really gave us a sense to think about these creatures, the extant creatures, in a new way, and basically showing that elements of that developmental program that build fingers and toes are probably present already in fins. The question is, we really don't know exactly what it's doing. We haven't done functional studies of this, nor do we know if it's independently regulated or not. So there's still a lot of questions, but it's there. It definitely is. But then what leads to the question is really, what is a limb, and how did it develop and evolve? And this is just a sort of take from Scott Gilbert's developmental biology textbook. Limbs are characterized by having two signaling centers, one called the AER and the other called the ZPA. There are a number of factors that are active in the limbs, which I'm not showing you. And indeed, there are a number of factors that have numerous feedback loops between the ZPA and AER. Just to give you a sense of what these are, let me show you the AER. The AER is a thickened ridge of tissue, and you see it here in this SCM, that sits at the distal end of the limb. Here you see a section of it. It's a pseudostratified epithelium. And it expresses a number of factors, most notably this factor FGF8. What's interesting about the AER is that if you cut it off at different stages of development, you get stage-specific truncations. That is, you cut it off early, you get a stump of a humerus, come it off later, and later you get more and more limb developing. So it's clearly permissive to limb development. The ZPA is a patch of tissue on the posterior side of the limb. It expresses a factor known as sonic hedgehog. If you misexpress sonic hedgehog or graft a patch of the ZPA to the opposite side of the limb, what you can do is get a mirror image duplication, as you see here in this chick wing. And there are a number of ways to do that. One way is actually to treat it with retinoic acid, beads. Another way is there are many ways to do it. The question they ask really is, how ancient are these signaling systems and the links between them? Are they characteristics of all appendages or just limbs? What we decided to do was then to look at the most basal of the animals with paired appendages on the cladogram, that is chondrichthyans. And so the idea being that does the common ancestor of all jawed fish have these signaling systems? And so we looked at sharks, skates, and rays. And this was work done by Randy Don in my lab. So here's what the AER in a limb looks like. With skate fins, we found the same thing. We found it has a thickened ridge of pseudostratified epithelium, and it has expression of FGF8. And when you manipulate it, you get limb-like effects. Do these things have a ZPA? This is what a chick limb looks like. There's the expression of sonic hedgehog. When you take a shark, a skate, or a ray, what you see is, similarly, you see a posterior patch of sonic hedgehog expression. What happens when you misexpress sonic hedgehog? Do you get a limb-like pattern? Well, what we did is we get these eggs of these things, and we can inject them with retinoic acid, which causes a number of effects. And what we did was we injected them with retinoic acid, as well as a number of other experiments. And here's the wild type. You see there's a posterior zone of expression here. And you actually upregulate sonic on the anterior side of the limb, as well as all the way along here, which is slightly different from limbs. But what's interesting is the morphological readout of this is that this is the untreated fin.